Okay, let's talk now about genetic mutations that can cause cancer. First of all, oncogenes. Oncogenes are formed when proto-oncogenes that promote cell division are improperly activated, and this may lead to an increased expression of the gene in a new location and the production of fusion proteins with new functions. So let's take a look at a RAS proto-oncogene. And we can see here that in cancer cells, the RAS gene product is locked into the GTP binding shape and does not require a signal at the receptor in order to stimulate cell division. So we see the normal cell and the cancer cell. And then here with proliferation, we see in response to growth factor binding at the receptor, the RAS gene product combines with GTP to promote cell division. Let's look at the movement of the proto-oncogene on chromosome 8 to the vicinity of a high active gene on chromosome 14, and this is going to cause Burkitt's lymphoma. So this would be a translocation. And remember that the translocation is one of the chromosomal mutations, one of the 5 chromosomal mutations. This is the Philadelphia chromosome, and this is found in patients with chronic myeloid leukemia, which causes a fusion protein to be made from a combination of genes on chromosomes 9 and 22. Okay, and moving on, let's talk about genetic mutations that can cause cancer. Tumor suppressor genes. One moment here. Okay, the next section of genetic mutations that can cause cancer is tumor suppressor genes, and these are genes that inhibit cell division. Genes that inhibit cell divisions are inactivated. Mutation in a gene that halts the cell cycle in G1 causes retinoblastoma. Mutation in p53, which is a gene that promotes apoptosis if a cell has damaged DNA, leads to a variety of cancers. And mutation in the BRCA1 gene is involved in tumor suppression and DNA repair, and this leads to inherited breast cancer. People prone to retinoblastoma have one mutated copy of the RB gene, which is RB negative, and one normal copy, which is RB positive. So a conversion of the RB positive copy to RB negative mutation leads to uncontrolled growth of retinal cells. In normal cells, the RB gene product controls the G1 to S transition. In normal cells, the p53 gene product acts as the G1 to S checkpoint, which prevents entry into the S phase if DNA is damaged. Here we can notice that the p21 inhibits intracellular signals that would activate EF2. And also notice that cells with damaged DNA do not pass the G1 to S checkpoint. And finally, it's important to know that in cancer cells, the mutated p53 gene product no longer stimulates p21 production. Cells will pass the G1 to S checkpoint even when the chromosomal damage exists. In normal cells, the p53 gene product stimulates apoptosis if DNA damage cannot be repaired. In cancer cells, a mutated p53 gene product no longer initiates self-destruction, and cells with damaged DNA can divide and more DNA damage can be accumulated. p53 is the most frequently mutated of all known cancer-causing genes, contributing to many types of cancer. So let's move on and look at DNA repair genes. These are genes that promote DNA repair. Genes that promote DNA repair are inactivated. So here we're talking about, for example, BRCA1 is a tumor suppressor involved in DNA repair, and faulty copies of BRCA1 cause inherited breast cancer. And the disease xeroderma pigmentosum results in a defect in excision repair. So we've talked about the gene mutations that can cause cancer. So, have you memorized all of this? We'll see.